Assalamu alaikum everyone. My name is Ahmed Eltayeb. I'm a second year medical student, and today I'll be giving you a lecture on the second week of embryonic development. If you have any questions, these are my contact details. Don't hesitate to contact me. For the objectives, we'll just go into each day in detail, and I'll mention a bit about extraembryonic mesoderm. Okay? So let's start. We have the uterus, fallopian tube, and the ovaries. Now, in the first week, we have fertilization. We have the zygote. Can anyone tell me what the zygote is? Okay, the zygote is basically a fertilized cell. And the zygote has mitotic divisions within the zygote. So you have 4 stages. You have the 2-cell stage, the 4-cell stage, the 8-cell stage, and the 16-cell stage. Now, the cells within the zygote, we call them blastomeres. And the blastomeres, they're loosely arranged in the first 2 cleavages. So in the 2-cell stage and the 4-cell stage, the blastomeres are very loosely arranged, right? But when it reaches the 8-cell stage, the blastomeres become very compact, right? And this is important because after the 4th cleavage, which goes from 8 cells to 16 cells, it's no longer a zygote. It becomes a morula, okay? A morula now has 2 distinct structures. It's not just blastomeres. It has 2 distinct structures, the inner cell mass and the outer cell mass. The inner cell mass, we call it the embryoblast. And the outer cell mass, we call it the trophoblast. Now, when the morula enters the uterine cavity, there are uterine secretions. And what is the function of these uterine secretions? They basically remove the zona pellucida. What does the zona pellucida do? The zona pellucida is responsible for protecting the zygote. So, as it travels along the fallopian tube, there are cilia, right? And if the cilia touches the zygote, then it will lead to damage, genetic problems, and a whole lot of problems later down the line, okay? So the zona pellucida is responsible for protecting the zygote from any physical damage, okay? When it reaches the uterine cavity, the morula needs to be inserted into the endometrial wall. So then we can start development. How does the zona pellucida come off? It comes off as a result of uterine secretions, okay? We need to remove the zona pellucida to begin implantation. Now, we said the trophoblast was the outer cell mass, and the embryoblast was the inner cell mass. Now, the trophoblast starts growing these finger-like processes, right? And starts digging into the epithelium of the endometrium. It starts digging, and then I forgot to mention, when the morula loses the zona pellucida, it's called the blastocyst. The structure is now a blastocyst. The blastocyst starts digging into the endometrium, starts digging through its epithelium, and it implants itself into the endometrium. Okay, let's begin day 8. Day 8 is when differentiation of the different cell masses actually starts happening. So, remember how we said the inner cell mass was the embryoblast and the outer cell mass was the trophoblast. The inner cell mass is now differentiated into 2 different layers. We have the epiblast and we have the hypoblast. Epi means above. So the epiblast is above the hypoblast, it's the one above, and it's adjacent to the trophoblast. It's right next to the trophoblast. The hypoblast, hypo means below. So the hypoblast is below the epiblast. And it's also adjacent to this cavity, which we now call the blastocystic cavity, okay? Just keep this in mind for later. What about the trophoblast? The trophoblast differentiates into 2 different layers. So we have this big, fat, multinucleated cell. We call it the syncytial trophoblast. The syncytial trophoblast, syncytium means network, okay? So the syncytial trophoblast is a network of cells. You can see there's no cell outlines, there's no clear cell membranes, and it's just one big fat multinucleated cell. So this is the syncytial trophoblast. The other layer, which has clear cell outlines, clear cell membranes, and they're mononucleated, we call it the cytotrophoblast. Cyto in German means comb. So you can see how it kind of resembles a comb. So yeah, we have the embryoblast differentiates into epiblast and hypoblast, epi being on top, hypo being below. And the trophoblast differentiates into two different layers, syncytial trophoblast, which is one big fat trophoblast, and cytotrophoblast, which are individual mononucleated cells. This is a more detailed diagram. So this is the epithelium of the endometrium, okay? You can see how the blastocyst has penetrated the epithelium of the endometrium. You can see how there's a big gaping hole at the bottom here in the epithelium. Now, the epiblast, we said, is above the hypoblast. But so on day 8, the epiblast begins proliferating. It begins proliferating, but it cannot move down. Why? Because there's a hypoblast below the epiblast. So the cells of the epiblast, they move up. They start proliferating up, and they attach, they make like a roof. They attach to the cytotrophoblast, okay? And then a cavity pops in the middle of these two layers. This is called the amniotic cavity. These cells, which are lining the amniotic cavity and are next to the cytotrophoblast, we call them amnioblasts, okay? And the cells that are lining the amniotic cavity, but are not adjacent to the cytotrophoblast, we call them epiblast, okay? A good distinguishing feature for the epiblast and the hypoblast is like the shape of their cells. So the epiblast looks like columnar epithelium, and hypoblast looks like cuboidal epithelium. Okay. This is everything I said, but in words, so you can always refer back to this later on. Okay, day 9. Day 9, we start seeing significant changes. So something I forgot to mention earlier, there's 2 significant landmarks that you need to be aware of. You have this part here, we call this the embryonic pole. And you have this part here, we call it the abembryonic pole. At the embryonic pole, we start seeing significant proliferation of the cytotrophoblast, okay? And you can also see the syncytial trophoblast starts getting bigger itself. So the syncytial trophoblast, I forgot to mention as well, doesn't actually undergo mitosis. It's the cytotrophoblast. The cytotrophoblast proliferates, and then its cells, it like squeezes upwards into the syncytial trophoblast, okay? So the cytotrophoblast at the embryonic pole, you can see it has more layers. It starts proliferating more, and then this contributes to the syncytial trophoblast and it makes it bigger, okay? There's also something really significant here as well. We start seeing holes, spaces in the syncytial trophoblast. Now, at first, these are just small empty holes, but then they start fusing together, and the fusion of these holes become lacunae. Lacunae basically means lake, okay, or lakes. So we start seeing these empty lakes in the syncytial trophoblast. This is why day 9 is often referred to as the lacunar stage. And something I forgot to mention. Something I forgot to mention. The epithelium here is closed off. It's closed off by a blood clot, which we call a fibrin coagulum. Okay, it's basically a plug that closes off the epithelium. Oh, sorry. And one more thing, really important. The hypoblast. The hypoblast starts proliferating on day 9, but it cannot move up because there's no epiblast. So the hypoblast starts proliferating, and then it moves down. It moves down and it forms a membrane adjacent to the cytotrophoblast. This membrane, we call it Huesner's membrane or the exocelomic membrane. Now this cavity, earlier we called it the blastocystic cavity, right? Now we call it the primitive yolk sac, okay? Or the exocelomic cavity.
Excuse me?
Yes?
I want to ask you to repeat part earlier. In the last slide.
Which part?
In this slide, but they didn't mention.
What do you want me to explain exactly? Hello?
Yeah, I'm with you, but they didn't specify on the chat.
Oh, are they asking me in chat?
Yeah. It was the other slide.
Should I just repeat the whole thing or?
Yeah, I guess.
Okay.
Just the last slide.
Okay. So, on day 9, we start seeing significant proliferation of the cytotrophoblast, okay? At the embryonic pole. The cytotrophoblast, you can see there's more layers here. Before it was single layered, now there you can see more layers. These layers, they start to push into the syncytial trophoblast, and the syncytial trophoblast becomes bigger itself, okay? We start seeing holes in the syncytial trophoblast. And these holes, they start to fuse and become bigger holes. And these holes, we call them lacunae, okay? So day 9, we often refer to it as the lacunar stage because we start seeing lacunae in the syncytial trophoblast. And these lacunae, they'll play an important role later on in the utero-placental circulation. We'll talk about it later. Something else you need to know here, the hypoblast. The hypoblast starts to proliferate on day 9 as well. It starts to proliferate and it makes a membrane. It goes down. Why does it go down? Because the epiblast is preventing it from proliferating upwards, okay? So the hypoblast starts proliferating down, and it makes a thin membrane which resembles simple squamous epithelium. This membrane, we call it Hussner's membrane or the exocelomic membrane. And one more thing, the epithelium here at the bottom, remember how it was opened up on day 8? It's now closed off on day 9 by a fibrin coagulum, which is basically a blood clot, and it acts as a plug. So it just plugs the hole in the epithelium. Okay. Question. Which of the following components play the most active role in invading the endometrium during blastocyst implantation? I'm just going to check. Yes, mashallah. The answer is C. So we said earlier, the syncytial trophoblast, the trophoblast starts growing finger-like processes, which it digs into the epithelium of the endometrium. And these finger-like processes, they come from the syncytial trophoblast. It's not actually the trophoblast, it's the more differentiated form, which is the syncytial trophoblast. Epiblast, amnioblast, these are the inner cell mass, so they don't really contribute to the digging. And extraembryonic somatic and visceral mesoderm, this is much later on, days 12, 13-ish. We'll start discussing it now. Okay. So on days 10 to 12, again, the cytotrophoblast still has lots of proliferation at the embryonic pole, okay? But there's something new about the lacunae. There's blood in them. So the syncytial trophoblast, it latches onto the capillaries, the maternal capillaries, and it degrades their walls. It destroys their walls, okay? As a result of the destruction of the walls, blood starts seeping through into the lacunae. Blood starts filling up the lakes, okay? And this is we call this utero-placental circulation because now maternal blood is entering the embryo, is entering the empty lakes in the syncytial trophoblast. Okay. Something else here is that so this is the exocelomic membrane or Hussner's membrane, right? And before, adjacent to the Hussner's membrane, we had the cytotrophoblast. But now there's something in between. What is this something? I think it's better demonstrated here. Yeah. So this something in between, we call it the extraembryonic mesoderm. The extraembryonic mesoderm there's holes start appearing in the extraembryonic mesoderm, okay? These holes are small at first, but then they start fusing, just like the lacunae. These holes, they start fusing and they become bigger, bigger, and bigger. And we call these holes the extraembryonic cavities, okay? Now, the extraembryonic cavities are important in separating the mesoderm into two layers, okay? So we have two layers of mesoderm, and in between we have the extraembryonic cavities. So we have the inner layer. The inner layer, we call it the extraembryonic splanchnic mesoderm. Another name for splanchnic is visceral. And whenever we say visceral, we're always talking about our internal organs, okay? So it makes sense that the extraembryonic splanchnic mesoderm is the inner layer of the extraembryonic mesoderm. And the somatic mesoderm, you can see it's adjacent to the cytotropoblast. The extraembryonic somatic mesoderm is the outer layer, okay? So inner, extraembryonic splanchnic, and outer, extraembryonic somatic mesoderm, okay? That's it for this slide. Now, day 13. Day 13, the cytotropoblast, it starts dividing even more, okay? Starts dividing even more and starts penetrating the syncytiotropoblast, and it's covered in syncytium, okay? These finger-like processes, we call them primary villi. Now, primary villi are important for the formation of the placenta. So this is the beginning of the formation of the placenta, but you don't need to worry about it in this lecture. You start to see more blood flowing into the lacunae. Okay. Now, remember the extraembryonic cavity? Look how small it was here, and look how huge it is here, okay? So we have three main cavities or holes we need to talk about. This one here is the extraembryonic cavity. So remember what I said? There were small holes appearing in the extraembryonic mesoderm. These holes were extraembryonic cavities. These extraembryonic cavities kept joining together, and made one big fat cavity, which we can see here, okay? The extraembryonic cavity is lined by the extraembryonic somatic mesoderm, okay? The extraembryonic cavity is lined by the extraembryonic somatic mesoderm. And just an important note, the extraembryonic somatic mesoderm and the cytotropoblast together, they make the chorionic plate. Okay, what about this structure here? This is the connecting stalk. Connecting stalk is basically extraembryonic mesoderm connecting the somatic and the splanchnic mesoderm. Okay, what about this cavity? And what's this hole here? It's better explained here. So remember this layer here? We called this the hypoblast. Why is it the hypoblast? Because the hypoblast is below the epiblast. Hypo means below. On day 13, the hypoblast begins proliferating downwards, because it can only proliferate downwards, right? It starts proliferating downwards, and it starts like strangulating this point here, okay? So imagine you have a balloon like this, shaped like this, and you strangulate the balloon at this point, at the lower portion. You'll have a small ball here, and you'll have a bigger one here, right? So this is essentially what's happening here. The hypoblast is proliferating, and the cells are moving down, and is strangulating at a certain point, and is forming like a small ball at the bottom, okay? And there's a bigger ball on top. So remember how earlier we called this the primitive yolk sac? When this small ball pinches off, we call it the definitive yolk sac, okay? Now, what is this small ball here? It's the exocoelomic cyst. So it pinches off from the definitive yolk sac, and it becomes its own thing, exocoelomic cyst. I'm not really sure about the function, but I don't think it's that significant. You don't need to really know about it. Something else you should know. So remember, the extraembryonic somatic mesoderm lines the extraembryonic cavity. What lines the definitive yolk sac? The extraembryonic splanchnic mesoderm. And the connection between the two is the connecting stalk, this thing here. The connecting stalk is just extraembryonic mesoderm, and later on, it becomes the umbilical cord, okay? Okay, if there's one message or one thing I want you to take home today, it's that the second week of development is known as the week of twos. So the tropoblast differentiates into two layers. You have the cytotropoblast and the syncytiotropoblast. The embryoblast, which is the inner cell mass, differentiates into two layers, the epiblast, which was on top, and the hypoblast, which was below. The extraembryonic mesoderm differentiates into two layers as a result of the extraembryonic cavities. So in between the cytotrophoblast and the Huesner's membrane, we had extraembryonic mesoderm. And then cavities started appearing and separated the two layers of the extraembryonic mesoderm. And this leads to the formation of two layers. The outer layer was somatic extraembryonic mesoderm, and the inner layer was extraembryonic splanchnic mesoderm. And two cavities are formed. You have the amniotic, so you had the epiblast. The epiblast started proliferating upwards, then a cavity popped out, and this is the amniotic cavity. And you have the yolk sac. The yolk sac in which Huesner's membrane was surrounding it, and when the hypoblast started proliferating downwards, forming Huesner's membrane, it also formed the yolk sac, the primitive yolk sac. And this is our last question for today. Between which two layers is the extraembryonic mesoderm located? Oh, wait. Can you please repeat day 12? Okay. Okay. This one, Ahmed? Okay, I'll just repeat it anyways. Okay. So on days 10 to 12, we have on days 10 to 12, remember the lacunae, how they were empty? So the cytotrophoblast starts to differentiate more, okay? It starts to proliferate more, and these cells go into the syncytiotrophoblast. The syncytiotrophoblast then grows bigger, and it latches onto the capillaries, the maternal capillaries, okay? These as the syncytiotrophoblast latches onto the maternal capillaries, it starts to degrade them, okay? The walls start to be destroyed. As the walls begin to be destroyed, the blood leaks into the lacunae. The blood leaks into the lakes of the syncytiotrophoblast. And this makes the uteroplacental circulation. Furthermore, we have, so in between the cytotrophoblast and Huesner's membrane, we have the formation of the extraembryonic mesoderm. The extraembryonic mesoderm starts to have holes in it. Just like the syncytiotrophoblast and the vacuoles, the extraembryonic mesoderm has holes in it. These holes start to fuse together and they become bigger, bigger, and bigger. As a result of these holes, two layers, you can see two distinct layers of the extraembryonic mesoderm. The inner layer, we call it the extraembryonic splanchnic mesoderm, and the outer layer, we call it the extraembryonic somatic mesoderm. Extraembryonic splanchnic mesoderm, splanchnic is another name for, visceral is another name for splanchnic, and visceral always means your internal organs. So it makes sense that extraembryonic splanchnic mesoderm is inside, it's the inner layer, okay? When exactly does the yolk sac start to form? Okay, when exactly does the yolk sac start to form before it divides to definitive and primitive? Okay, so in the beginning, let's see. In the beginning, we had here. In the beginning, we had before differentiation, for everything, we had the embryoblast and we had the trophoblast. The embryoblast is the inner cell mass and the trophoblast is the outer cell mass, okay? So this cavity here, this cavity that is next to the embryoblast or the inner cell mass is the blastocystic cavity, okay? Now, the primitive yolk sac forms when the hypoblast proliferates downwards, okay? So we see the hypoblast is on day 9, it starts to divide rapidly and it proliferates downwards, okay? it forms a membrane called Huesner's membrane. And the membrane, this membrane is the lining for the primitive yolk sac. So day 9 is when the primitive yolk sac is formed, okay? Then you have the definitive yolk sac. The definitive yolk sac, one second. The definitive yolk sac is formed on day 13 when the hypoblast starts to proliferate down here, starts to proliferate down, the cells start to move down, and then there's a point at which the cells start to strangulate, like a certain point. It starts to make like a dip, and it starts to strangulate this point, okay? Then when this point closes off, this small ball, it buds off, it forms the exocoelomic cyst, okay? And this, what's remaining here, this is the definitive yolk sac. So blastocystic cavity is there from the beginning. Primitive yolk sac is there on day 9 when Huesner's membrane is formed, and definitive yolk sac is there on day 13 when the hypoblast proliferates and forms 2 different structures, the exocoelomic cyst and the definitive yolk sac. I hope that answered your question. Can you repeat connecting stalk and the chorion? Okay. Okay. So the connecting stalk here is it's a bunch of extraembryonic mesoderm, which connects the extraembryonic splanchnic and extraembryonic somatic mesoderm, okay? Just a little fun fact, I don't think you need to know, but the connecting stalk later on becomes the umbilical cord, okay? So the connecting stalk connects extraembryonic splanchnic and extraembryonic somatic mesoderm. It's like the bridge between the two. And the chorionic plate is basically extraembryonic somatic mesoderm plus the cytotrophoblast. These two combined makes the chorionic plate. And the chorionic plate is one of the membranes for your placenta, which you don't need to worry about in this lecture. What is lining of primitive yolk sac? The primitive yolk sac, it's lined by Huesner's membrane. So we go back to day 9. We have the hypoblast. The hypoblast begins to proliferate, which means the cells divide rapidly. The hypoblast begins to proliferate, and then the cells, they go down. They go down because they cannot go up because of the epiblast, right? So they go down, and they make a membrane of simple squamous epithelium, and we call this membrane Huesner's membrane. And Huesner's membrane, or the exocoelomic membrane, is what lines the primitive yolk sac. I think that's it. Does anyone have any more questions? Oh, wait, Oh, wait, my bad. Sorry, guys. Okay. Between which 2 layers is the extraembryonic mesoderm located? The answer is E. The exocoelomic membrane is also known as Huesner's membrane, okay? So between Huesner's membrane and the cytotrophoblast, we have the extraembryonic mesoderm, okay? I'll just show you here. Here. So here, you have the simple squamous lining. And here, you have the cytotrophoblast. And in between, you have this structure. We call it the extraembryonic mesoderm, okay? So the extraembryonic mesoderm sits between the Huesner's membrane, or the exocoelomic membrane, and the cytotrophoblast. So that's why the answer is E. Does anyone have any questions regarding the question, or even about the lecture?